So how are you all?
Good, Miss Nandi, and you?
Good. Just tired.
Yeah, me too.
Just came back from campus. Okay, so what's the time?
It's time to start.
Okay, let's start.
Mhm.
Okay, so I think I talked up to these, right? This was the last slide. Um, I talked on Monday. Okay, so the glycocalyx capsule slime layer. Monday I already talked about it, um, um, and even I talked about Fimbri and pili. So glycocalyx, that's the outermost layer of prokaryotic cell. Not all the bacteria has that glycocalyx. Okay, there are few bacteria who carry these capsules or the slime layers, but not all the bacteria has capsules and slime layers. So glycocalyx, that's the jelly-like substance, surrounding the cell wall of prokaryotic cell. Two different types, capsules and slime layers. When it's irregularly arranged, okay, and or you can say loosely arranged, then it's called slime layer. When the arrangement is solid and regular, then it's called capsule. Um, both capsules and slime layer help prokaryotes to attach on any surface. Okay, this is their protective structure, and at the same time it determines the virulence, or you can say the pathogenicity of the bacteria. The bacteria with the capsules and slime layers, it's very difficult to kill them. Okay, versus bacteria without any uh, capsule or slime layer, um, it's very easy to kill them. Fimbri pili, they are the outermost appendages actually. Um, both almost look similar. Both are hair-like structure, but Fimbri short in length. Pili, they're long, slender hair-like structure. Both are adhesive organ for the prokaryotic cell. They help prokaryotic cell to be attached on any surface, solid surface. Um, and at the same time, they are the virulent factor, pathogenic factor for that prokaryotes. Pili, there are some pili, not all, but there are few pilis. It's called sex pili. Um, that sex pili or sex pili, they are the reproductive organ. Um, you can see in this picture, it's almost like sexual reproduction in prokaryotes. It's not really sexual reproduction, but it's almost like, we can say that it's almost like sexual reproduction, because here we need two organisms. Okay, basically two organisms take part here. Not only that, their strains are completely different. Okay, just like our male and female, they don't have male female, but they have different strains. Okay, there are few characteristics that actually determine the bacterial strains. So what happened, the opposite strains of the prokaryotes, they come in close contact with each other, and then they make a connection with the pili or the pili or the pillars. It's a tube like structure, and then they exchange their genetic material. Okay, so basically these two organisms or the prokaryotes, we marked them as positive strain and the negative strain. The prokaryotes with extra chromosomal material, basically positive one, actually the donor, and basically it donates the genetic material, or you can say the DNA. Negative strain, they are the receiver. They receive that genetic material, okay, and they exchange that DNA or genetic material through this pili. So that's how this pili actually help them in reproduction. You can say it's a kind of reproduction, or you can say that when they exchange their genetic material to increase their genetic variety, okay, or variance. So that's the function of the pili. Flagella, prokaryotic flagella, it's very, very, very characteristic features. Okay, it has three fundamental structure. One, that's it's called basal body. Basal body remains attached with the plasma membrane. Okay, so this is the cell. If this is the plasma membrane, let's say this is the plasma membrane, then outside bacteria has one layer, cell wall, right? So this is the, let's say this layer is the cell wall. This basal body remain inserted inside the plasma membrane, not inside the cell wall. Okay, that's a very, very characteristic features of prokaryotic flagella. If you examine the eukaryotic flagella, the origin and the attachment is little different, and the arrangement is little different than the prokaryotic flagella. Anyway, the bacterial or archaeal flagella, this basal body, it works as engine. Okay, this is like you can say the motor. So this is the motor of this locomotory organ. That day I told, I hope you remember, that flagella work as a locomotory organ. Okay, it helps prokaryotes to move from one area to another area. So this basal body is the engine of this locomotory organ. Then the end part, the furthest part, that's the filament. And in between this filament and motor, means this filament and motor remains attached with a hook. This part, this is loosely attached both with the filament and the basal body. So these are the three parts of flagella. Now what happened, this basal body, it actually helps to move the filament, okay? And by movement of the filament, these prokaryotes, they actually move from one place to another place. Now, based on the arrangement of flagella, so usually most of the time they have just one flagella, but there are different bacteria. They have so many different types of different numbers of flagella. So when there is only one flagella, just like this picture, just one flagella, and at the one end of the body, that arrangement is called monotrichus. Okay. Amphitrichus, there are two flagella or maybe two tuft of flagella. Okay, instead of one, just one, it could be just one tuft, means multiple flagella, but at one point, one origin, they have just one root. So anyway, if those flagella are arranged on both the end, both the surface or both end of the body, then that's the amphitrichus flagella. Lophotrichus is all the flagella are at one end of the body. Basically, there is one tuft only at the one end of the body. So now the difference between amphitrichus and lophotrichus. Amphitrichus, there could be just two flagella on both the end of the body, or it could be, like at one end there are multiple flagella. It's a tuft when they have only one origin. So it could look like even that. So so that picture or this, they are the amphitrichus, but just only one tuft of flagella at one end, that's the lophotrichus. Peritrichus means the whole body is surrounded with multiple flagella. So that's the peritrichus. Now the prokaryotic movement, how flagella, or you can say how by moving the flagella, they can move from one place to another. At first, why do they need to move? You can ask me that, what caused them to move? It could be the attraction or repulsion of any chemical. Okay. It could be the adverse environment. It could be the food. Okay, because of the scarcity of the food, they need to move from one place to another place. So all these things are reason so that they need to move from one place to another place. Most of the time they are inside the water or little bit of humid area, but they even can stay on the solid surface, but these flagella help them to move. Now when they move, only two direction they can go. Either they can move straight, okay, like as a straight line. When they move following a straight line, then it's called run. Okay. Or they can just make a circle like this. To change the direction, they actually make a circle. Okay, and this movement is called tumbling. So there are two ways they can move. One is running or tumbling. Now by run, they can go straight. By tumbling, they actually move in a circle, but they only do tumbling when they want to change the direction. So basically what happens, let's say over here you sprayed that bleach. Okay, so this is the area of the bleach on your workstation. So they need to move away from this area. So what they will do run, and the other thing is when they do run, they move their flagella anti-clockwise. Okay, counterclockwise or anti-clockwise, they move their flagella to go straight. So what they do in case of peritrichus flagella, all the flagella gather at one place, and then they very quickly move that tuft of flagella anti-clockwise, and by that they can just go straight. So if they start moving from here, they are going, let's say, this direction, and over here we have bleach again. So what they do, they want to go that side. Okay, so if they want to come here, what they will do, at this position, they will start rotating by tumbling. Now, how they can change their movement? By, at this moment, they rotate their flagella clockwise. Okay, so suddenly they change the movement of the flagella. They start rotating their flagella clockwise, and they will start taking turn, and then at certain point they will just started moving their flagella anti-clockwise again, and they will go straight. So that's how they move every time. What they do, so run, short run, then tumbling, then run again, then tumbling. It's very funny and very interesting to look under microscope their movement. So we are done with the prokaryotic cell organelles. Now the eukaryotic cell organelles. Eukaryotic cell, we have both plant cells, animal cells. Okay, so for the animal cell, animal cell don't have the cell wall, but the plant cell, yes, we have cell wall. For the fungi, they have cell wall. So mostly for the eukaryote, they few of the eukaryotes, they have cell wall. Few of the eukaryotes, they don't have cell wall. But for all eukaryotic cells, we have plasma membrane, okay, or the cell membrane. Same structure like prokaryotic plasma membrane, biphosphate bilayer, and in between the phosphate bilayer, we have protein. Okay, different types of protein, and it's a fluid mosaic structure because of the presence of that cholesterol and unsaturated fatty acids, the fatty acid king, in one glycerol fatty acid leg. So that's why it looks like it's fluid. Basically, the protein, they float in between the phospholipid. So that's the plasma membrane structure. Exactly same, not exactly, but almost same as prokaryotic cell membrane. The cell wall, it's different, like fungi. For the fungi, or the fungus, the cell wall is made up of chitin. Okay, very characteristic feature. There are no other organism who has chitin cell wall. Plant, they have cellulose cell wall. Okay, so it differs, okay, from one organism to another organism. So if we go, if we start looking from the plasma membrane, so immediately beneath the plasma membrane, we have cytosol or the cytoplasm. Okay, the brain of the cell is the nucleus. Almost every time it remains at the center of the cell, and we call the nucleus as the brain because it controls most of the activity, chemical activities, you can say, inside the cell. We have not all eukaryotic organism, but there are few eukaryotic organism who has that flagella. There are cilia. So many different type of this type of organelles are there. We will when I think it's on chapter 5, when we are going to work on the eukaryotic cells, specifically different type of cells, like when we are going to study about the protozoa, when we are going to study about the protists, fungi, then we will work on all these appendages, about flagellum, about cilia. Okay, so now in this chapter we will only work on the common eukaryotic double membrane organelles, okay, cell organelles. So the first one is the nucleus. So nucleus, double membrane structure, again that lipid bilayer, phospholipid bilayer. But over here, for in case of nucleus, these two bilayer, they're contagious, means they're basically completely attached with each other, but there are two lipid membranes are there. Okay, so basically in between two membranes, nothing is there. They're tightly attached with each other. Now this lipid bilayer, or that double membrane, in between these two membrane, we have small pores. Okay, and we call that one as nuclear pore. So that nuclear membrane, the membrane or the bilayer, it's also called nuclear envelope. Okay, so this is the envelope, and inside the envelope we have nuclear pores. Okay, now the purpose of the pore is, remember I told that a nucleus is the actually it's kind of brain. So lots of stuffs need to go inside the nucleus, and lots of stuff needs to go outside, or you can see inside the cell from the nucleus. So all the materials, you can see different types of ion, different type of sugar, large carbohydrates. Not only that, even ribosomal RNA, rRNA, messenger RNA during the protein synthesis, just before protein synthesis, rRNA moves through all these pores. Okay, messenger RNA, they move through these pores. Okay, so uh, that's the purpose of all these nuclear pores. Now, attached with the nuclear envelope, at you can see there is a dark stained structure. Okay, a little harder area, and it's called nuclear lamina. Nuclear lamina, nothing but the collection of microtubules. It is made up with microtubules. Okay, so basically skeleton system. So one of the endoskeleton is microtubules. Mostly this nuclear lamina, they are made up with mostly with the microtubules. So the function of my nuclear lamina is to give the shape of the nucleus. Okay, it provides the rigidity, endoskeleton structure, you can say. So that's the purpose of nuclear lamina. Inside the nucleus, the jelly-like substance is just like cytoplasm. We have nucleoplasm. We inside the nucleus, we can find the DNA, or you can say the chromosome. Now the chromosome, before cell division, or you can say almost all that time, they remain associated with the protein. Okay, so and when cell is resting, when cell is not dividing, so all these chromosomes, they remain associated with the protein. Okay, so and really we cannot count the chromosome number in this situation, and we cannot see or understand the shape of the DNA. When this is the situation, means this DNA or the chromosome, they are very thin and loosely arranged and associated with the protein, and we cannot see the shape, we cannot count them. Okay, so then we actually call that DNA and protein together is the chromatin material. So basically this chromatin material is nothing but the association of DNA and protein. Okay, and for eukaryotes, our protein that remains associated with the DNA is called histone protein. Okay, make sure you remember that eukaryote has histone protein, but prokaryote, they don't have that histone protein. So that's the chromatin granule. Nucleolus, very important. At one spot inside the nucleus, it's really dark stained, okay, circular area. That area, that spot is called the nucleolus. This nucleolus is associated with, or you can say that responsible for this rRNA synthesis. So this part is very important because that rRNA or ribosomal RNA that is actually going to be synthesized inside this area. How or from where, we really do not have idea. So many things are undiscovered still now. Okay, so we don't know. Recently, we just discovered that this is the spot responsible for rRNA synthesis. Now, that ribosomal RNA or rRNA is very, very important because they are responsible to make the ribosome, to construct the ribosome, okay? So rRNA and protein together, they make that large subunit and small subunit of ribosome immediately just before the protein synthesis. When inside the cell, you can see the ribosome, that means this is the time either the protein is going to be synthesized, or you know, it's going on, or very shortly cell is going to make the protein. So otherwise ribosome, once the protein synthesis is done, ribosome get dissolved. So this rRNA is very, very important. Endoplasmic reticulum. So these are the endoplasmic reticulum. Endoplasmic reticulum, you know, like this is the system of endoskeleton, or you can say the endomembrane system. That's very characteristic features for eukaryotic cells. Okay, so it's called endomembrane system. This endomembrane system means all these membranes are surrounded by membrane. Okay, and that's the one characteristics of endomembrane system. Other characteristic is all this membrane actually originated from the plasma membrane of cell. Okay, so most of the time, so if this is the cell, if this is the nucleus, all this endomembrane system originated from the plasma membrane. So that's another characteristic features of endomembrane system. So all of the endomembrane systems origin actually got from that plasma membrane. So one of that endomembrane system is endoplasmic reticulum. So when we studied that endosymbiotic theory, scientists believe basically this plasma membrane, they got you know, invaginated, and ultimately they get separated from the plasma membrane, and all this, you know, small portion of the plasma membrane, they took different shape, and ultimately in different condition. Like if this membrane surrounds the matrix of mitochondria, then finally they got converted into mitochondria. So that's how all these different type of organelles actually originated. So this endoplasmic reticulum, what happens? So this is the cell, and from the cell, this membrane, first it got invaginated, then in second step, this endomembrane get separated from the plasma membrane and remain attached with the nucleus. Okay, and they get invaginated, or you can say they get folded. Okay, so that's the origin of endoplasmic reticulum. Endoplasmic reticulum, it's the interconnected array of, you can say, the tubules or flattened sac or disc-like structure. Either they are tubules, tube-like structure, flattened either sacs or flattened disc-like structure. Okay, but all of them are internally connected with each other. Okay. Um, and every time that endoplasmic reticulum, you are going to see they remain attached with the nucleus. Okay, that endoplasmic reticulum and Golgi over here in this picture, they look almost similar. Only difference how you are going to identify is this Golgi or Golgi body, they remains little far away from the nucleus, but the endoplasmic reticulum, they are always remain attached with the nucleus, or you can see them immediately outside of the nucleus. Okay, so that's the difference between ER and Golgi. This is the Golgi apparatus. Both of them look almost alike, you know. Golgi also, they has tube, they have vesicle, they have flattened sacs. So, but endoplasmic reticulum always remain attached with the, or you can say immediately outside of the nucleus. That's how you can identify that this is the endoplasmic reticulum. Now, endoplasmic reticulum, there are two different types, either rough endoplasmic reticulum or smooth endoplasmic reticulum. So if you see the picture, this is the smooth ear and this is the rough ear. Okay, we also call endoplasmic reticulum as ear. So either rough ear or smooth ear. So only difference is between these two is outside of that rough endoplasmic reticulum, you can see that small dot-like structure. They are the ribosomes. Okay, so rough endoplasmic reticulum carry ribosome, okay, on its surface. Smooth endoplasmic reticulum, they don't have, or you can say they don't carry any ribosome. So that's the structural difference. Now the function, we know that ribosome is responsible for protein synthesis, right? So this rough endoplasmic reticulum helps in protein synthesis. Very, very important point. Very, very important questions. The difference between the function of rough endoplasmic reticulum and smooth endoplasmic reticulum. Okay, very important, and make sure you remember that. So rough endoplasmic reticulum, they help in protein synthesis. They carry protein all the way to Golgi apparatus. Okay, basically this rough endoplasmic reticulum, when ribosome make the protein, then that protein needs to be transported all the way to the Golgi apparatus. Okay, because this is the processing center of the protein. So from here to here, this endoplasmic reticulum actually carry all this protein. So this rough ear, they help in protein synthesis. Smooth endoplasmic reticulum, they are responsible for carbohydrate metabolism. Okay, that's the number one function. Number two function, they are responsible for lipid biosynthesis. So this is the place where carbohydrate metabolism takes place. This is the place where cell make extra lipids. Number three, very, very important function, that is detoxification. Okay, so any toxic product is actually going to be detoxified by this smooth endoplasmic reticulum. So this smooth endoplasmic reticulum are very, very important. So make sure you memorize that the difference between the smooth and rough endoplasmic reticulum and their function. Now the Golgi apparatus. So Golgi, again, they are made up with either the tubules or sac or flattened disc-like structure, and these sacs are called cisternae. When these flattened disc or sac-like structure are inside the Golgi, they have special name. We call this cisternae. This is actually the protein modification and processing center. So that's the function of the Golgi. So protein processing and modification center. Basically, what happens here? Like, let's say this protein. So from the ribosome, from here it goes over here, and it has specific direction of that Golgi's, all the cisternae. One corner is called cis, and one corner is called tart. So they have specific direction. Anyway, so when that protein reaches over here, now that Golgi, it actually modify, purify, or maybe it make it large. Sometimes, like glycoprotein, what happens? So glycoprotein means it is the combination of glycogen, means it's the carbohydrate plus protein, right? So these things, the production of glycoprotein, it actually occurs inside the Golgi apparatus or Golgi apparatus. Okay, so basically when this protein reaches over there, then Golgi apparatus adapt that glycogen or carbohydrate, monosaccharides or disaccharide, and then gives that complex protein structure. Okay, so that's the it's that's how it is process or modify the protein. So that's the function of Golgi apparatus. Make sure to study the structure of all cell organelles, and very, very important is you really need to know the function of each cell organelles. Cytoskeleton system, that's the internal network supporting system, or you can say that it's a internal skeleton system. Eukaryote has three different type of cytoskeleton: microfilaments, intermediate filaments, and microtubules. These are the three different type. Okay, so microfilament, they are made up with actin filament. Mostly the cytoskeleton, as I told you, that they are the endoskeleton of cells. So basically they give the structure. They are the support system of the cell, and the third function is the transport material. Very important function of cytoskeleton. So support, they provide structure, and transport. So actin filament we can find in the microfilament, and that microtubule is made up with tubulin. These two are very, very important. Where do we find actin filaments? Where can we find the tubulin filaments? Okay, so remember that actin filament we can find inside the microfilament, and microfilaments, it is a type of cytoskeleton. Tubulin we can find inside the microtubule, cytoskeleton microtubules. Okay, these microtubules, they are responsible for cell division. Okay, remember that during the cell division, that centrosome, they make that fiber, right? That actually pulls away, separates away the chromosome materials, or you can say, yeah, they are responsible. Actually, they are responsible for the cell division. So how they help? These tubulin granules, they actually make centrosome. That's another cell organelles. We are not actually sure at this moment, but yes, you can consider it as a cell organelles. This each centrosome has two centriole, and both the centriole are arranged, they are 90-degree apart from each other. And there are 9 sets of 3 microtubule. So 3 microtubule arranged in 9 sets. So basically, altogether 27 microtubules are there in one centrosome. And they actually make the spindle during the cell division. That's the function of centrosome. Now the mitochondria, powerhouse of cell. All the cellular respiration takes place inside the mitochondria. Very, very important. Cellular, when we are talking about the cellular respiration, then basically we are talking about the production of oxygen, and that production of oxygen takes place over here inside the mitochondria. Not only that, plus ATP. Okay, so this is the site of oxygen and ATP production. So mitochondria, double membrane structure, external membrane and internal membrane. Now the internal membrane is highly folded. You can see these things. This is the internal membrane. So basically, one external membrane and internal membrane is highly folded, so to increase the surface area. You can understand, like if this much long a straight line, if you fold it, then it will be like this. So it will occupy very short area, right? So short distance. So that's the reason to increase the surface area, this inner membrane is highly folded, and this folding of all this inner membrane is called cristae. And this is the place where most of the ATP production occurs. Okay. Now, inside these two membrane, outer membrane and inner membrane, we have jelly-like cytoplasm, or you can say mitochondrial cytoplasm. It is called matrix. We don't call cytoplasm, but it's mitochondrial cytoplasm, and it has special name. It's called matrix. Remember that mitochondria, they are actually a prokaryotic bacteria, right? So that's the reason to differentiate from our cytoplasm. We give that the mitochondrial cytoplasm a special name, and that is the matrix. Inside the matrix, they have their own DNA. They have their own ribosome. Very, very important. No other organelles has that except mitochondria and chloroplast inside the plant. They have their own DNA, own ribosome, and this DNA, they divide independently. They are not dependent on our nuclear chromosome or hereditary material, okay? So, and in all these, the cristae, like the inner membrane and the matrix, all carry so many different type of enzyme for the cellular respiration. So these two areas are very hot metabolic place. Okay. Chloroplast inside the plant, we don't have, animal cells do not have. And this chloroplast, few of the prokaryotes, they have chlorophyll-like structure, but that chlorophyll is not associated with the chloroplasts, okay? So this plastid or chloroplastid, again, double membrane structure. All the organelles of eukaryotic cells all has double membrane structure. So outer membrane, inner membrane, okay? there is a space in between that outer membrane and the inner membrane that also has jelly-like structure. I forgot to tell you over here also. In between that outer membrane and inner membrane, we have a little bit of jelly-like structure. Okay. So same over here, inside the chloroplast. Now, the cytoplasm of chloroplast, it's called stroma. Okay, so don't forget that the cytoplasm of mitochondria is called matrix. Cytoplasm of chloroplast is called stroma. Cytoplasm of any eukaryotic cell we call cytoplasm or cytosol. Okay, so the stroma, that's the semi-liquid jelly-like structure. Stroma carry the structure which is called granum. So basically inside the double membrane of chloroplast we can find out two things. Okay, either granum and the stroma. So stroma is the cytoplasm that actually carry that granum. So what is granum? Granum is actually the stack of thylakoid. So basically a few flattened disc-shaped structure together is called granum. Okay, and each flattened sac is called thyla or thylakoid. Okay, so basically, so one on top of other one, upright position, all those flattened discs together make the granum, and individual discs are called thylakoid. So both this granum and stroma together make the inside of the chloroplast, and inside this thylakoid membrane we can find out that chlorophyll. Okay, and the plant use that chlorophyll to absorb that sunlight. Cell membrane transport. So I already talked about when a membrane transport, we already talked about the diffusion and osmosis. Okay, active transport, passive transport, I already talked. I didn't talk about when or how membrane transport large particles. Okay, when they actually transport large particle from outside to inside or inside to outside. That process is called phagocytosis. Okay, so phago means solid particles. So when they transport any large solid particles, that's called phagocytosis. In that case, what happened? The plasma membrane invaginated, they make a sac-like structure, they surrounds that solid particles, and ultimately they make a sac-like structure, and that sac get separated from the plasma membrane. So they creates a small vacuole. Okay, and ultimately when that vacuole goes inside, lysosome is there who has enzyme, and they dissolve the plasma membrane, and that substance actually comes out of that vacuole. So this process is called phagocytosis. When it is liquid, large amount of liquid, it brings from inside to outside or outside to inside, then that process is called pinocytosis. Pino means water. There are another transport mechanism, it is called receptor-mediated endocytosis. Endo means when they are taking in. So two things are there. Endocytosis, large particle, when they move large particle, there are endocytosis and exocytosis. So endo means in, so when the particles are coming inside the cell. Exocytosis, when the particles are transported out of the cells. That's the exocytosis. So in this case, what happened outside of the plasma membrane, we have receptors, okay? And all these receptors has different shape. So any particles that needs to come in, that particle's shape needs to match with the receptor. If that receptor fit inside the particles receptors, then that membrane actually allow that receptors to bring that particles inside. So basically in this case, large particles, especially the ion, sugar is very large molecule. So sugar, different types of ions, they cannot actually directly come through the membrane. In this case, they take help of the receptors, or you can say that they take help the protein compounds inside the cell. So basically these receptors needs to be matched or fit with those molecules, and then it helps that molecule to come inside the cell. And this process is called receptor mediated endocytosis, and it is endocytosis because it's coming in. So this happens, this exocytosis, endocytosis happens in case of large particles, large molecules. So that's the membrane, membrane transport. Okay, so we are actually done chapter 3. Okay, do you have any question on chapter 3? This chapter is very easy. Lots of information that you have to memorize, but it's easy to understand. Let me start chapter 4. What happened? Okay, so that's our chapter 4. Chapter 4 talks about the prokaryotic diversity. So basically, it talks about the bacterial prokaryotic diversity, their structure, and we are going to work on the classification. Okay, we are going to talk about the different types of prokaryotic cell. So general characteristics of prokaryote. Remember in first chapter we talked about it that prokaryotes, they are ubiquitous, means they are everywhere. You can find them everywhere. At the ocean floor, melted ice, inside the melted ice, in high altitude, inside the volcano, inside the acid, inside the boiling water, inside the refrigerator, like Listeria, one bacteria, it's called, sorry, I have to, okay. So it's called Listeria. They can, they remain survive inside the freezer for more than a month. Okay, so month after month, actually, the Listeria. So you can understand that you can find them everywhere. All these bacteria. Basically, with these slides, we want to talk about the diversification of different types of bacteria. You know how extremely diversified all these prokaryotes are. That's the purpose of this slide, the first slide of this PowerPoint. So over here, some prokaryotes called halophiles. Hello, the word hello means high amount of salt. Okay, philes means where they live in, or you can say that they are the lover, or where they actually live. Okay, so halophiles means the salt lover, means this group of bacteria, to survive they need high amount of salt. Okay, so that's why they are called halophiles bacteria. One example, don't forget, very important, every day we are going to work with this specimen in lab. It's called Staphylococcus, Staphylococcus epidermidis. The Staphylococcus epidermidis, they live on your skin, okay, upper surface of your skin. So our skin is a little bit salty, right? Um, and the staphylococcus, they need that salty environment. Okay, so these staphylococcus epidermis, they are also called the halophiles. Okay, so halophiles, they can thrive in extremely salty environments such as the Dead Sea. Inside Dead Sea, it's really salty. It's not like it's not like regular ocean or regular sea what we see. Last year, I went to Turkey. I saw that it's so heavy, that salt, it's so condensed, the water. Okay, very little amount of oxygen. Difficult to survive for any organism except the halophiles. So even over there inside the Dead Sea, you can find out the bacteria. Here the archaea, the Holobacterium salinerum, shown here in this picture, is the halophiles that lives in the Dead Sea over here. Now, this Halobacterium salinerum, they are photosynthetic archaea. They are not bacteria. Okay, they are archaea. So they are archaean. And they are actually photosynthetic. They can do photosynthetic synthesis. And when they do photosynthesis, what they do, they actually create light pinkish or purple chemicals they produce. Okay, and that's the reason, do you see over here, they looks like, dark purple or pink, room kind of, they look like. So that this is nothing but the product of photosynthesis. The bacterium Sivanella, this bacteria, and over here, this bacteria lives in the deep sea, where there is little oxygen diffused in the water, and these bacteria, they are able to survive in this hard environment. Okay, in the deep sea means extremely high amount of salt and very low oxygen. They can survive, but Sivanella, they need oxygen. It's not like that they do not need oxygen. So over there inside the Dead Sea, it's high amount of carbon dioxide and carbon monoxide, very little amount of oxygen. Okay, but Sivanella needs oxygen, but very little, but still they need. So what they do, they have small flagella-like structure. You can see, we called them as nanocables. Okay, so they move that nanocables. Okay, and they generate electricity inside that ocean water, sea water. Okay, and scientists are working to produce electricity by using that Sivanella. These nanocables, it's nothing but you can say the sensory organ. Okay, so sensory organ means it actually helps to find out the oxygen for the bacteria. So they actually moves that appendages or that nanocables to go towards the oxygen inside that deep sea. Okay, but because of the movement of that appendages, it produces electricity. So basically, we are now at this moment, scientists are behind the cyanula to produce bioelectricity. So basically this slide talks about the the diversification of the prokaryote, both the archaea and the bacteria. Importance of prokaryote, key points. So how important they are very, very important. Mostly they are helpful. Okay. Yeah, few of the important features we already talked in chapter 1. Here, few other importance of prokaryotes we are going to talk. So first, the decomposers. Okay, in that chapter also we talked about it, that there are few chemoheterotrophs. You can say chemoheterotrophs. This group of bacteria, they actually are the decomposers. So basically not all the prokaryotes, some of the chemoheterotrophs. What they do, they worked on the dead animals, dead plants, animal litter, plant litters, animal wastes. Okay, they break down the large organic. So basically our body is made up of organic compound, right? So when we die or plant dies, this group of bacteria, they worked on those large organic compound. They break down that large organic compound into small chemicals, like you can say carbon, nitrogen, okay, so phosphorus. So they release all these different type of chemical elements inside, in the environment, and that actually helps very much. They help in carbon fixation, like animals like us, we cannot absorb the inorganic carbon. Like in the atmosphere we have huge amount of carbon dioxide, right? But can we just get that carbon dioxide? Can we utilize that carbon dioxide or carbon directly from the air? No, we cannot. We are responsible, we are actually dependent for carbon onto plants. So plant, they can directly absorb that carbon dioxide from the environment. That carbon dioxide is inorganic carbon. Okay, so only plant can absorb that inorganic carbon in the form of carbon dioxide from the air during photosynthesis. Okay, and we animals, when we eat the plant product, from all those plant food, we are going to get the organic carbon. Okay, so this decomposer actually helps to break down that carbon. They help to make ammonia, like that organic organic substances. They release the nitrogen. They help that nitrogen to convert into ammonia, and that ammonia is very, very important for the plant's health. Okay, so there are so many different ways, like whatever I told. By that way, all these decomposers, they actually enrich the environment with lots of different type of chemicals. If they do not do that, then we are going to lose all these chemicals. Necessary part of soil formation, all these prokaryotes, by decomposing or by making the biofilm, they actually make the soil. Okay, they enrich the soil. That's very important. Encompasses the origins of Okay, I can soil formation. Another way uh, they are very important. The bacteria, what they do, there are. I hope you have heard about that the leguminous plant. Okay, that leguminous plant, like you can say alfalfa. Okay, these plants are the leguminous plants. This is the leguminous plant. So you can see all these nodes over there. We have special type of um, mm, prokaryotes, bacteria over there. What they do, they absorb carbohydrate and acid from those roots of that leguminous plant, and they convert those carbohydrates and acid into humus. It's called humus. And that humus, actually, that enrich the soil. That's the part of the soil that creates the soil. So these are the few procedures how important the bacteria are for soil formation. Encompasses the origins of metabolism and metabolic diversity. These bacteria, they are very, very resilient, very, very adaptive, very, very adaptive. They can mutate within few seconds. Okay, so whenever they needed, they can change even their metabolic activity. Like us, we actually cannot change our metabolic activity. Now, if you like to drink acid, you cannot, because you really do not have any enzyme inside your body that can help you to digest that acid. Okay, so we have a very fixed mechanism. We cannot change our metabolic activity, but these bacteria, they frequently can change their metabolic activity, and that's the reason they are so adaptive. You spray antibiotic, within few days or few months, they will be resistant to that uh, chemicals. Okay, so they can frequently change that metabolic activity. And nowadays, because of they are so flexible in their metabolic activity, scientists, we are actually using like cyanobacteria. We are using cyanobacteria. They are very, very flexible when it comes about the metabolism. By using that cyanobacteria, we are actually experimenting to produce biofuel, because we saw that if we change, we want to change the regular metabolic pathway of cyanobacteria, they actually can do it. Okay, and instead of producing that fatty aldehyde, they can produce that biofuels, and scientists are researching on that. Origin of photosynthesis and formation of atmospheric oxygen. I talked about it during first chapter, that a huge amount of oxygen is produced by the prokaryotes, especially the the photosynthetic bacteria like cyanobacteria. There are purple green bacteria, sulfur bacteria. So they actually produce huge amount of oxygen. Still, more oxygen is produced by the bacteria than the rainforest, Amazon rainforest. Okay, so total Amazon rainforest, how much oxygen we get from that Amazon rainforest, we get more oxygen still now from the prokaryotes. Okay, now they produce really huge amount of oxygen. Essential components of symbiosis. Symbiosis means it's a very close relationship between two organisms, and to do a symbiotic relationship, these two organisms has to one organism, either it should be in or inside the other organism. It's really close contact in between. Okay, so if you are like let's say inside your house, you have family members, so you are not you don't have symbiotic relationship with your family members, your husband or your child, that's not the symbiotic relationship. It has to be really close interaction, and one organism has to be either in or on. Okay, so inside my body, lots of bacteria are there, or outside of my skin, lots of different types of bacteria are there. That's the symbiotic relationship, because all those organisms, they are living inside my body and on my body. Okay, so that's the symbiotic relationship. And it has to be between two different biological species. Again, because it has to be two different biological species, you and your spouse, you are not symbionts for each other, okay, because you are the same biological species, you are not two different biological species. That's the other reason. So these are the two conditions to fulfill that symbiotic relationship. There are so many different type of symbiotic relationship. Mutualism, okay. So remember, every time when we are talking about the symbiotic relationship, we are actually talking about two different organisms, okay. So in case of mutualism, what happened, both the organisms are benefited, nobody is harmed, okay. And nobody is inactive, you can say inactive. Both are benefited. Example like E. coli and human. E. coli or Escherichia coli, that's a gram-negative bacteria. They live inside our gut. Okay, what they do, they help us to absorb vitamin E, vitamin D during the digestion. Okay, they help us to absorb. If we don't have E. coli inside our body, then we cannot absorb vitamin E, vitamin D. So in this case, what's happening? The human are highly benefited, okay, from the E. coli, and E. coli, they get a place inside our body to stay, to live in. They are going to get the food, whatever we are absorbing, producing from the digestion, you know, they are going to get their food from there, from our gut. So both the organisms are benefited. So that's why the relationship between this specific group of E. coli, we have few E. coli who are pathogen. We are not talking about all E. coli, but one specific strain of E. coli who are actually who lives who actually share the mutualistic relationship with the human. Okay, so remember this one, not all E. coli. There are E. coli who are pathogen. So that's an example of mutualism. Amensalism is one is harmed, one organism is affected and harmed, other organism is unaffected. Okay, so over here we gave example of Staphylococcus aureus and Lucilia sericata. So now this Lucilia sericata, it's a bug, one type of bug. Okay, Staphylococcus aureus, that's a gram-positive bacteria, and they lives on your skin. And at that Lucilia sericata, sometimes they lives on your skin too. So that Lucilia, what they do, their larvae, they lay eggs on your skin. So they're very microscopic organism, and their eggs are microscopic. So that's the reason you cannot see them. So that Lucilia sericata, they lay eggs on your skin, and that eggs develop into larvae, and that larvae is very poisonous to this Staphylococcus aureus. So on your skin, Staphylococcus aureus is very pathogenic for us. Okay, if that Staphylococcus aureus from your skin, if they get entry inside your body, they are very harmful. So this Lucilia larvae actually secretes one special type of enzyme that kills this Staphylococcus aureus. Okay, nowadays we are trying to use this Lucilia sericata larvae, to our dead tissue. Sometimes it happens like, for the debridement, like we have necrotic tissues, that is the tissue because of the infections and all, because of the Staphylococcus aureus infections and other bacterial infections. There is no medications. Okay, so we have to just ampute that organ of your body. We have, maybe it's your hand, so we have to cut that out. So nowadays we are using that Lucilia larva to eat all those dead tissue from your body. Okay, so there is no medication, and instead of cutting that part out, we are spraying that Lucilia larva, which is going to eat, digest all those dead tissue. Okay, and that actually cures that part of your body. So that's still under experimentation, but we are working on that. So the relationship between the Staph aureus and Lucilia, it's unaffected, nothing had happened to Lucilia, but Staphylococcus aureus, they are going to be killed by that larva. So over here, this group of organisms, they are harmed, and Lucilia, they are unaffected. Okay, very interesting. So that's the amensalism, that relationship. Commensalism, one is benefited. So amensalism, one is harmed and one was unaffected, right? Commensalism, one is benefited and one's remain unaffected. Easy example, Staphylococcus epidermidis and human. That I told you that Staphylococcus, they always, every time, millions of Staphylococcus epidermidis, they reside on your skin because they are the halophiles. They need a little bit of little more salt concentration. So they live on your skin. Usually they are harmless when they are on your skin. So they eat all of your dry skin, they eat, they suck the carbohydrates and all those. So basically they get the place to stay and they get food from your body, mostly the dead skins. And they don't harm unless they can go inside our body. As long as they are outside on our skin, we are unaffected. So relationship between the Staphylococcus epidermidis and human is called commensalism. Parasitism, one organism is benefited, highly benefited, and another organism is harmed. So over here, I gave you one example of mutualism where one strain, make sure you every time remember that one specific strain of E. coli who has mutualistic relationship with human. Other strain of E. coli who are very, very harmful, they're pathogenic for us. Okay, so those group of those strains of E. coli, they gives us so many different types of disease. They gives us diarrhea. They are responsible for urinary tract infections. Okay, so human are harmed, and E. coli, they can stay, they can get food, so they are benefited. So that relationship is called parasitism. Now the taxonomy of prokaryotes. So I told you that this chapter we are mainly going to work on the classification, okay, of prokaryotes. Very important, and from your textbook it's very confusing. Okay, very, very confusing. How they actually classified. So I tried to make it easy for you to understand. Okay. So the whole group of prokaryotes. First thing, we are going to divide these prokaryotes into two groups, two domains. Okay, one is bacteria and other is archaea. That you already know. Okay, so bacteria and archaea. In this chapter, we are mostly going to work on the bacteria because the archaea, they are not that much diversified, okay? What happened? So mostly we are going to work on the domain bacteria. Now bacteria, we have two different groups, gram-negative bacteria and gram-positive bacteria. We worked on that based on the cell wall, okay, based on the cell wall thickness or based on the presence of peptidoglycan, you know, how thick the peptidoglycan layer is. Based on that, we have gram-negative bacteria, gram-positive bacteria. Now, gram-negative bacteria, there are two different types, proteobacteria, non-proteobacteria. Okay. Now, these proteobacteria, so gram-negative and proteobacteria are subdivided into 5 groups: alpha, beta, gamma, delta, and epsilon proteobacteria. Okay. Non-proteobacteria, we have 3 different groups: Spirochetes, CFB group, and Planktomycetes. Okay, so that's a proteobacteria, non-proteobacteria. So completely, this part is completely up to this is gram-negative bacteria. Now the gram-positive bacteria, we have low G plus C and we have high G plus C. So G plus C, I hope you remember from the AP one, like the DNA structure, that DNA nucleotide sequences. I hope you remember that adenine, guanine, cytosine, thymine. So over here, to classify that gram-positive bacteria, we are actually going to work on the guanine and cytosine percentage. Okay, how much percentage of guanine and cytosine are there? So based on that, they are in 2 groups. So more than 50% guanine cytosine, they are the high G plus C, and less than 50% guanine cytosine, they are the low G plus C. Okay, so up to this is very easy, right? So gram-negative bacteria, either proteo, non-proteo, gram-positive bacteria, low GC, high GC. Now the phototrophic bacteria, we have mostly gram-negative. We have few gram-positive bacteria who, and also we have archaea who are photosynthetic bacteria, who actually can do photosynthetic. So this group of bacteria, they are both on gram-negative and gram-positive bacteria. Okay. So proteobacteria. So basically we are going to work on this group. Okay. So who are actually proteobacteria? So we can understand that gram-negative bacteria, it's a huge group of bacteria. They are completely divided into proteo and non-proteobacteria. So how we divide them, on what basis? So basis is actually by RNA sequencing. Okay, based on the RNA sequencing, RNA structure, RNA sequencing, scientist Carl Woese, he actually proposed that there is a big, huge group of bacteria, and they are called purple bacteria. So he told that all the purple bacteria and their relatives needs to be placed in a separate phylum, because they are closely related with each other by RNA sequencing. So that RNA sequencing is actually separating this group of bacteria with this group of bacteria. All the bacteria, means all these alpha, beta, gamma, delta, epsilon, all these bacteria, if we do RNA sequencing, they have relationship. Okay, close relationship. But this group of bacteria, that Spirochetes, CFB group, Planktomycetes, this group of bacteria, their RNA sequencing is little different than this group. Okay. So Carl Woese told the purple bacteria because of their color and their relative, and then later we actually gave them this name, proteobacteria and non-proteobacteria. How we actually choose this name? Because of the Greek god Proteus. You know, Proteus who could assume many shapes. Okay. So basically, this group of bacteria, proteobacteria, they are very, very diversified, and that's why we actually, based on that Greek god's name, Proteus, we chose this name, proteobacteria. So alpha, beta, gamma, epsilon, 5 groups are there. Okay, it's almost 7 o'clock. Let's keep today up to this. Okay. So Monday, I think that's the Labor Day. We don't have any class, but Wednesday we are going to start from here, alpha proteobacteria. Okay. So do you have any question? No? Brandy, do you have any question? No? Okay, no questions. Good. Okay, so let's end today over here. Happy Labor Day weekend. Enjoy your Labor Day. And Monday, we are not going to have any Zoom session, but I'm going to see you on Wednesday. Okay. So next Wednesday, we'll be done this chapter 4.
Miss Nandy, for those of us who have lab next week, we don't go to lab on Monday, right?
No. Next week,
Okay.
there is no lab. No lab next week. Both Monday, so basically Monday, Wednesday, Tuesday, Thursday, there is no lab. No lab.
Oh, okay.
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 So next week, holiday. Enjoy your holiday. Only Wednesday, we will meet for a little bit of time for this chapter.
Okay. Thank you.
Okay. Okay. Good night.
Good night.
Bye.
Bye.
Okay, you said next week there's no lab for Monday and Wednesday.
Hmm?
You said next week there's no lab for Monday and Wednesday.
No, not during the Labor Day weekend. After that, again, we are going to start the regular schedule.
All right, thank you.
You're welcome. Good night, Shinala.